and then once Maggie's done the count, she can hit record, the red bar will show, and take it away. Thank you.
Awesome. Thanks, Patricia. I have the great pleasure of introducing today's speaker, Dr. Ellen Damshan. When the PCA committee was meeting to talk about speakers and her name came up, I was super excited because although she probably doesn't remember me, I remember her from working on my PhD at University of Florida, and particularly her work in longleaf pine communities at the Savannah River site in South Carolina. Dr. Damschen is a full professor at the University of Wisconsin-Madison in the Department of Integrative Biology. She researches patterns of species diversity, what causes them, and the impacts of global change on biodiversity loss. To me, what's really remarkable about her work is both the scale of her experiments, how large scale they are over space, and also the time spans studied. And I think that her work provides really unique data that can help first determine what's driving species declines, and also, and very importantly, the strategies that we can use to promote species persistence and recovery. So I am really eager to learn how over a decade of research on conservation corridors has evolved and changed, and particularly about how it can help inform our knowledge of species diversity. So with that, I'm very happy to turn it over to Dr. Ellen Damshan.
Thank you so much, Anne. That's a really kind introduction. Can you give me a thumbs up if this all looks okay and sounds okay? Okay, great. So I'm really happy to be here. This is an unusual opportunity, and I've been really excited about it because I think that the partnerships between land managers and researchers are really critical, and oftentimes we don't have enough communication between those two groups. And I'm just going to say from the outset that I think the work that I have done is successful because we have really strong collaborations with our federal agency partners and other management agencies at the state and private even level. And because of those relationships that we have, we're able to ask important questions, but that have real, hopefully, conservation context that's applicable. I'll just put a question out there that you can sit with throughout the talk. At the end of the talk, I am really interested in getting perspectives from managers in terms of what questions need to be addressed. So I'll be sharing some of the things that I have worked on, but if there's anything related to the things that I've worked on that you think are really important moving forward in the next 5 years or so, I would love to have you put those questions in the chat or something at the end so that I can get a handle on what you're facing in your agencies right now. So today,
Dr. Damshan, I'm sorry, this is Anne again. I'm sorry to interrupt so rudely, but I only see a static version of your video, and if that's on purpose, that's totally fine. But I thought before I could actually see you moving. I just wanted to clarify if.
No, hang on one second.
Sure. Is that happening for anybody else? Now I see you.
Okay, I can't see myself. So, oops, now I can, okay, hang on one second.
Now you're moving and you look great.
Great, so. That's good. My talk is not moving, so let's try that. Okay, here we go. So the talk that I think was advertised was the role of habitat corridors and plant diversity, and I'm definitely talking about that today. I'm also going to broaden it out to just talking about landscape influences, because sometimes it's a corridor or a thin strip of habitat connecting to otherwise isolated reserves. But sometimes it's just general connectivity on the landscape. And when we move outside of an experimental setting, that's oftentimes what we're thinking about. And so I'll try to start with the experimental results, and then I'll be moving to more realistic landscapes. We're thinking about connectivity more broadly and looking at the relative influences of local and landscape processes on plant community dynamics, and especially under climate change and in a fragmented landscape. Just before I begin, I'm at the University of Wisconsin-Madison, as Anne already said, but I do a lot of work in the southeastern United States and more broadly, and my interests really came from thinking about biodiversity as a whole. And so what this is showing happens to be bird diversity at a global scale, and ecologists often wonder about what it is that causes diversity to range so widely across the globe. And this is just showing sort of a latitudinal gradient of diversity where we have more species near the tropics and fewer near the poles. There's a lot of attention given to diversity at these very large scales. We also, though, can ask questions at smaller scales. And so probably like many of you, this is something I tackle frequently. I'll go into the field. This is one of my field sites in longleaf pine, Savannah, and put down a meter square quadrat. And a key question is why in one site do I have maybe 60 species in a quadrat, and in another site I have 0 species in a quadrat? This is really the process of two types of factors. So what we see in a local site is both the result of the local environment, so things like soils or other plant competitors or other species interactions, and then also at a broader scale, landscape factors, so movement of individuals from place to place. And it's the relative balance of movement to a site and then local processes at that site that determine the total number of species and their composition at a local site. Now,
Dr.
this
Damschen,
is, yes.
Dr. I'm so sorry I keep interrupting you. I'm only seeing your first slide, and I have a feeling you've moved on.
Okay, I have.
Okay.
So I do see you're on the landscape movement, local environment slide.
Oh, do you see that, Patricia? Okay.
Yeah.
I just stopped sharing for a second. I'm gonna just redo this.
You see her slides? Okay. I'm sorry.
Seems like most of us see the slides changing. It's
okay. It's
Yeah.
just me. I see it now. I will not interrupt anymore, and I will leave it to Patricia.
Please do. Yeah, please do if you see anything that's not working on your end.
Okay.
But sometimes Teams just freezes up, so it's good to just try to close your background stuff in.
Okay, thank you.
Okay, so we have these, sort of two classes of processes that determine local species diversity. Now, amidst all of this, and this is, of course, what you were just talking about at a very large picture, is the loss of biodiversity at a global scale. And I'm just circling in white here. This is a report that came out from IPBES, which is the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services. This report came out in 2019. And has a lot of really important information, including sort of the percentage of species that have been lost globally by different taxa. And I've circled plants. So we have ferns and relatives and monocots on the upper left, conifers and dicots towards the bottom, and then cycads at the bottom, which is actually one of the most threatened groups of species worldwide. And so this is just all to say that plants are endangered, just like animals are, and we need to be thinking about the factors that are causing their loss just as much as animals. This is, of course, in large part due to our widespread impact at the global level. So this is a photograph you may have seen before. This is from space, just showing lights at night across the globe. And humans have a pervasive impact on our environments across the world. This is in large part due to climate change, which is represented by that image in the upper left, showing average temperatures in parts per million of CO2 going up over time. Changes in fire regime, the loss and rearrangement of habitat. And then in the bottom right-hand panel, there is also a loss of fire in addition to the increase in wildfire that we know so much about from the West and Australia and recent events. I'm also going to put up a figure here that in many ecosystem types, including prairies and savannas, it's the loss of fire that is causing declines of understory species. And so this is called musification. So this is just showing over time essentially the amount of burned area has decreased over time. So this is a problem for many ecosystems and is different depending on the ecosystem's fire regime. So it certainly wouldn't be applicable to Western stand replacing fire regimes. So on top of these two classes of processes, we have global changes that are influencing both landscape level movement and the local conditions. And so understanding how they're affecting these two classes of processes and their relative importance is important, but not just the kind of problems that these human-induced factors have caused, but also solutions. So the bottom right-hand photo here is the Yellowstone to Yukon Corridor Initiative, which is trying to connect habitat throughout the western part of North America. And corridors, which I'll talk about today, are one conservation strategy that we could implement to try to protect biodiversity. In addition, restoration is a key component of these solutions. So we can think about both positive and potentially negative impacts of humans. So today what I'm going to do is I'm going to have three parts to my talk. I'm just going to first start by telling you a little bit about the approach and study systems that my lab works in, just to give you some context of, who I am and what we're thinking about. And then I'll address two questions. The first question I'll address is, how does landscape connectivity affect long-term community dynamics? And this will be mainly from an experiment that we have now had running for 22 years. And then the second question that I'll address is, how do landscape and local factors affect long-term change? And that's where I'll try to broaden it out to think outside of that experimental context into, quote, real landscapes. I want to just acknowledge from the start that when I say I or we, I am talking about a whole collection of people. It includes a lot of people that are in my lab, which are many of them are represented here over time, and also agency partners. So for example, we work a lot with the Forest Service at Savannah River site. We work with the Department of Defense at Fort Bragg and Fort Stewart on the eastern United States. We work with the Natural Resource Conservation Service in Wisconsin. We have had funding with one of my postdocs with USDA NEFA. So there's a lot of partners that have been really important in this work. So to give you some background, the work that goes on in my lab really works at a very large scale. So sometimes that might be experiments, which I'll talk about today, that are manipulated at a landscape scale. Other times it might be having a simple experiment that is replicated across space. So some other work that I'll briefly touch on at the end has been done across space and across the longleaf pine range. In addition, we do work that is long-term. So I've used historic data, both from Wisconsin and the Western United States in the Klamath-Siskiyou region, to look at long-term change over 50 to 70 years. We also have long-term data sets. So I'll talk today about 18 years of data from an experiment. And then finally, we're really interested in something called functional traits, or the function of species. It's essentially characteristics of species, in this case plants, that can be measured, that give you some indication of how they respond to particular ecological processes. So for example, you could look at the dispersal mode of a species as if it's dispersed by birds or wind, and think about how good of a mover that species is, and that could be a categorical functional trait of that plant. Study systems we've worked in are all grassland and savanna ecosystems. So I'm just kind of giving a quick snapshot of that. I'm not going to talk about all of these today, but we work in tallgrass prairie and oak savanna in the Midwest. I've done work in the Klamath Mountains, like I mentioned, in serpentine grasslands. I've done some work in the Ozarks and Dolomite glades. And then I have done a lot of work in longleaf pine savanna and the southeastern United States. And all of these systems have something in common. They, first of all, have been maintained by low intensity, frequent fire and or water limitation or nutrient limitation. And so many of these ecosystems have a fire return interval on the order of 1 to 5 years. So routine low intensity fire is what these communities were evolved under, and we have changed those regimes in many cases to have a lack of fire. So I'll start with this first question. How does landscape connectivity affect long-term community dynamics? So this question arose because I was really interested in thinking about the effects of habitat loss and fragmentation as a new graduate student many, many years ago. This paper had come out by Wilkov et al. In 1998, and it showed that over 90% of the species on the endangered species list were there because of habitat destruction and degradation. And many other factors like non-native species and pollution, overexploitation, and disease are also important, but by far and away, habitat destruction and degradation was the most important factor. I think now we'd probably add on climate change, and there's been some recent analyses that suggest that that's also a really important factor. And typically, all of these others are interacting with habitat loss and fragmentation to affect species loss. Now, fragmented landscapes have three general characteristics. The first thing that happens in a fragmented landscape is there's a decrease in total area. So larger habitats become smaller. The second thing that happens is that as habitats break up into smaller pieces, there's an increased prevalence of edges. So if you were to measure the perimeter of the figure on the left and divide it by its total area, and then look at this one on the right, which has the same amount of area but broken into pieces and took its perimeter, you would have a larger edge to area ratio. And then finally, there's increased isolation. So habitats that are normally closer together become farther apart. So there's greater interpatch distance. And all of these factors are known to affect species, but separating out the independent effects of area, edge effects, and isolation is really tricky, especially in realistic natural landscapes. So one conservation solution that has been proposed to mitigate the negative effects of habitat fragmentation is the use of landscape corridors. So you just take a piece of habitat that's linear and you connect these otherwise isolated habitat patches. And corridors have been hypothesized to increase movement, population sizes, genetic diversity, and species diversity. We have a lot of data now about movement rates. We have some, but not a lot, on population sizes. We have hardly any on genetic diversity, and we have some on species diversity. So there's been a lot of work that's been done, but a lot left to go. Corridors were proposed in the 1970s and quickly adopted. And so here is just a smattering of images of where you can find corridors in conservation plans or actually on the ground. And for a long time, their use kind of exceeded the evidence that we had about how and whether corridors worked. Most of the evidence that was really kind of concrete and was able to isolate the role of isolation or reconnection was done in small-scale experiments. And so this is really important work that was done in Marcel Holyoake's lab, Andy Gonzalez's lab, and Sharon Collinge's lab, looking at species diversity in either test tubes with microorganisms, moss arthropods in these very tightly controlled chambers. And then Sharon's work in Grasslands was looking at insects in California. So the question that we set out to ask was, do corridors work at larger spatial scales? Because while that work was really important and tested some really important questions, it was not done at the scale of working landscapes. And so in the winter of 1999-2000, my collaborators created a landscape that is at the Savannah River site in South Carolina. At South Carolina, we have 8 replicate sites that each look like one of these photographs that has a collection of 5 patches. So we have a center patch that is 100 by 100 meters or 1 hectare that's connected to another equally sized patch with a 150 meter long corridor that's 25 meters wide. We also have a winged patch that has the area of the corridor added onto either side of it in the shape of the corridor. And what this patch does is it controls for area. So this patch, this patch, the winged patch, is the same area as the connected patch, but is unconnected. It also has the same edge to area ratio. The unconnected rectangle is the same area as the patch. It takes the area of the corridor and tacks it onto the back of a 100 by 100 meter patch, but also is unconnected. So one of the things we can do is compare these different patch types to find out if, for example, edge effects are important in driving the pattern that we observe, or so we can compare rectangle to either winged or connected to get at that. Or we can try to isolate the effect of connectivity, by just comparing the connected patch to winged patches. In some cases, which is the case in the results I'll show you today, there's no difference in the unconnected patch. And so we will combine those together and just compare the connected patch to both of the other unconnected patch types, winged and rectangle. The surrounding pine forest is the habitat matrix. And at first you might think, oh, well, there's no difference. But if you look in the understory of this pine plantation matrix, it's very densely planted and there's not much going on in the understory. In the patches, however, when that's opened up, tree density is low, and we also use routine fire to maintain these patches. We end up with a rich herbaceous understory. And as I mentioned, this is in the longleaf pine ecosystem, and so these openings are actually the more appropriate historic habitat that managers are interested in. And so sometimes I'll get the question of, isn't this the inverse of habitat fragmentation? But that mainly comes from an assumption that we're talking about forested ecosystems. And in this case, longleaf pine is a woodland ecosystem that has very sparse pine trees with this very, very rich herbaceous understory. So many of the species of conservation concern from longleaf pine are found more often in inside of our patches than they are in the surrounding habitat matrix. One other thing I should mention is that this ecosystem type, for those who aren't familiar with it, is a biodiversity hotspot. There are actually more species at small spatial scales than even in the tropics. So you can go to a 1 by 1 meter quadrat and you can have up to 60 species in that quadrat at small, fine spatial scales. And it's also associated with numerous threatened and endangered species, including the red cockaded woodpecker, the gopher tortoise, and indigo snake. So what we're doing here is measuring a number of plant species. We call that plant species richness, and we're doing that in each of our patches. And so I have this photograph here to show that the data that I'm going to present today are all just from the areas highlighted in yellow. We're not including the center patch. And that's important because it means that the area of these patches is remaining constant, and the only thing that's changing is the connection or the shape. When we go out each summer, and I actually just completed this survey mid-June this year, myself and my colleagues, especially Lars Brodvig, who's at Michigan State University, we start in one corner of a patch and we walk back and forth, guiding ourselves by these tall PVC grid poles, visually surveying the entire area of the patch. So this is essentially a census of all the plant species in the patch. This work was started in the summer of 2000. We did a pilot study that year as we were getting going, and so we didn't sample the whole patch in this first year, but then we did thereafter. And the important thing by these first initial bars is that there's no difference between the yellow and blue. So the connected patches are yellow, and both of the unconnected patches, the winged and the rectangle, are in blue, and there's no difference. And this is important because it means that our experimental landscapes weren't laid on top of some pre-existing biodiversity gradient that we weren't aware of, and we are able then to see the effects of these landscape manipulations over time. The next years we did these full censuses, so that's why there's a jump up in the total number of species. But what we're really interested in is the difference between these bars. And so this was work that I published in 2006, so it's a long time ago now. But one of the key findings here was that if you plot that difference over time, there was an increase in the connected patches in species richness relative to the unconnected patches. And so this is the first experimental evidence demonstrating that the role of corridors through connectivity per se, not through area or edge effects, leads to an increase in the number of plant species. A key question after this, we found this result that then led to this long-term work was what's going to happen next. And so we wrote a grant where we outlined some long-term predictions. So again, on the left is the same y-axis. It's the difference in species richness between connected and unconnected patches. And then I have time on the x-axis. And so one prediction we made was that we'd see that initial increase that we saw through 2006. And maybe it would go up for some amount of time, but then at some point it would plateau. So the relative benefits of corridors would reach their maximum, and then we just have a high level of species richness relative to unconnected patches over time. But the second prediction could be that maybe the spatial scale of these landscapes relative to the organisms that use them, in this case plants, would allow all plants to colonize any patch, but just over some longer time period, and unconnected patches could catch up. And if we saw that, then the difference that we initially saw might actually drop off over time, and all patches would have the same richness. And so we would be back to kind of square one. So these are the two predictions that we made. And so then we collected data for a long time. And after 18 years, we decided to retest this. So here again is that difference on the Y and time over 18 years on the X. And this is what we found. And this is using an occupancy modeling approach that we worked on with Rob Fletcher at University of Florida. And I want to just say a couple things about what you're looking at. The first is that this is a per species estimate of the for each species. We're creating the likelihood that it's found in a particular patch type. And so what that means is that we have occupancy models for almost 300 species, and then we're taking the net sum of all of those 300 species. And that's going to be important for some of the next findings I'm going to show you. The other thing is that because this is based on a binomial distribution, the line can appear curved when it's actually a linear relationship, and that's what we were interested in testing. And so we ran some analyses to assess whether the shape of this line was straight or a linear fit, or whether it was some sort of curved line, to see which of our predictions were matched. And this bottom inset shows that the models that were the best fit by far and away were ones that were linear. And so this didn't conform to either of our predictions. It was a surprising result. Maybe we haven't had enough time for this to play out yet, but at least over almost 20 years, we are seeing a continual increase in the benefits of corridors over time. So a key question is, why is this happening? So we think a lot about the likelihood of species arriving or colonizing sites, and also the likelihood that they'll go extinct in conservation. So what I'm going to show is two rates: the colonization rate, and again, this is a per species per year rate over time, and the extinction rate. So this is the colonization rate. So what this is showing is that it's the difference between connected and unconnected patches. So we have a 5% greater likelihood on average that a species will colonize a connected patch relative to an unconnected patch, and that's a per species per year rate. So that might not seem like a lot, but it's like compound interest in a bank, where you have an interest rate that accrues every year. And so if I were instead to say, I'll give you a bank account with a 5% interest rate, you might think, oh, that's a great interest rate. And so that's kind of how to think about it here. Similarly, with the extinction rate, those rates drop in connected patches. So on average, per species per year, there's a 2 to 3% drop in the extinction rate. And both of these are continual over time. So we have not seen this rate change very much for either of these over time. So a question that we wanted to ask is, if you put in a corridor, what kind of temporal benefit do you get out of this? How does it change management potentially when you're thinking about managing on the order of years? And so what we did is we looked at the cumulative colonization probability per species. So what this is going to look at is, on the y-axis, it's saying at a particular time, what's the likelihood that a species will colonize a connected patch or an unconnected patch? So we could take 0.5, which is 50%, as just an example, and say, what's the likelihood that a species has a 50% chance of being in a connected patch or an unconnected patch? And so I'm going to put these up here, and this is the 50% mark that I was just talking about, and we can plot these lines then for connected and unconnected. And if we see where that crosses this 50% line, this is just arbitrary, you could do it anywhere you wanted, and you draw that down, what this is showing is that in patches that are connected by a corridor, the time for colonization of those patches is reduced by 2 years relative to an unconnected patch. And if you could, because you can do this at any point, like I could say, what's the likelihood at 75% for the likelihood of a species being present in one of these patches is 75%. It's on, because you can do that over this range of probabilities, it varies from 2 to 7 years difference in the kind of temporal scale of dynamics that are playing out in connected versus unconnected patches. And I think that that's actually kind of astounding that you could kind of get a jump on things at that rate by having connections between patches. So a key to this is, why is this happening? We know that extinction rates and colonization rates are affected. One of the reasons theoretically that corridors should work is because they alter movement rates. So we have done some additional work looking at the role of dispersal. So I'm just going to share a few highlights of that to talk about the mechanisms. My colleagues Doug Levy and Josh Tewksbury worked a lot on bird dispersed seeds. And so this is an example showing bird dispersed fruits, wax myrtle and upon holly with the eastern bluebird. And what they did was they put fruits in this center patch that were marked with a fluorescent dye powder. And then they had traps that looked like this photo on the right. And they were able to collect fruits from these traps and see if the fluorescent dye powder was present or not. So this is showing the results of that. So we have, I'm going to start with the bottom one here. This is the proportion of fluorescent fruits in connected, winged, and rectangular patches. And you can see that the proportion fluorescent is higher significantly than either in the connected patch than either of the unconnected patches. And in this case, this species was not present in the patch in any of our patches. And so we didn't need to mark it with fluorescent dye powder. So this is just the number of seeds per patch. And again, it was higher in the connected patches than in the unconnected patches. So this shows that corridors affect bird movement. And we know that that's because there's additional studies that Doug and Josh have done where they found that it affects bird behavior. So birds tend to forage in the center of these openings where fruits are abundant, and then they move to the edges, they perch for a minute and bounce off that edge and come into the center of the patch. So with that behavior, they essentially bounce their way down the corridor to another connected patch. We also looked at wind dispersed plants, and to do this, we first measured wind itself. So we put up these very tall towers and we measured the wind speed and direction in our landscapes at these locations with the black dots. Because we have done this work at Savannah River Site, we have a national lab there that measures all sorts of abiotic things, but including wind very high up above the canopy. So they have towers on site that are above the canopy. And so we know what the wind is doing in general in this upper left-hand panel. And then we can measure what's happening in our landscapes. So this is a wind rose, and the bars represent the proportion of movements of wind in that particular direction. So what you can immediately see is that corridors tend to funnel wind down them. They move in line with the corridor, and wings do that to some degree as well. Inside the centers of patches, it seems to be more uniform and perhaps more in line with what's happening above the canopy. And then the color of these bars shows the speed. And so you'll see that yellow and purple are much faster wind speeds. So one other thing that happens inside of a corridor is that the wind speeds up, and same with the wings to some degree. We also know from additional modeling work that uplift is also more common in connected patches. And uplift is really important for wind dispersed seeds. Many have adaptations to only be released from the plant when wind is coming from the ground and up, which makes a lot of sense. And so we knew that wind was likely changing the movement of plants. To get at that, we marked some seeds that were fake, but they glowed in the dark. And we put them in these craft boxes. My postdoc, Dirk Baker, made this whole apparatus. And these seeds that are fake, they actually fall at the same velocity as many of the native species in our system. So we got a team of people and we put them at all of the different locations that were in black dots in the previous graphs, and had them get on the radio and say, all right, we're going to start releasing seeds every 30 seconds for the next half hour. And so they did that. And then after nightfall, we could go back with a black light and we could find these seeds and map them. And this is important because these seeds were released then under the same meteorological conditions, and we could look at how far they went in each of these locations. So these are long distance movements, which means that they went farther than what a typical ballistic or just gravity model of dispersal would predict. And what it's showing here is that seeds that were released from the mouth of the corridor were more in line with the corridor, and also a greater proportion went longer distances in line with the corridor. That didn't happen so much with any of the other patch types, including in the wing, but it sort of demonstrates that the changes that we're seeing in wind dynamics actually translate to what's happening to wind dispersed seeds. Now, to kind of put the icing on the cake, John Orrock, one of my collaborators, came up with an idea using the wind data from Savannah River site. We could figure out how much wind was blowing in line with the corridor over time. So in our landscapes, this was published in 2014. So from 2000 to the time that these data were collected, we knew how many species were in each patch, and we also could make this calculation of how much wind had blown down the corridor during that time period. So we calculated every time the wind blew within 30 degrees of a particular landscape. And one thing I didn't mention is that we have these 8 landscapes, but the corridor is aligned differently in every landscape. So we could do this for each landscape, no matter what the orientation of the corridor. And that here on the x-axis is this mean corridor aligned wind speed. And we plotted that against the species richness of wind dispersed plants. And I was really surprised, but we got this really strong relationship where the more wind and the faster it's blowing there is in a connected patch, the more species we see. So translating that then to real landscapes, quote unquote, we have these experimental landscapes. They are very large scale for a study of its kind, on the order of hectares. But when thinking about management, and I'm sure you might be thinking about this if you're working with real landscapes, it's still small. And so there's some other work that we've done that tries to get at the role of connectivity in landscapes that are not experimental. So one result, I'm just going to present a few results from other systems to talk a little bit about this. This system is in Wisconsin. I'm at UW-Madison, and we have the great fortune of having a historic data set that was collected in the 1950s by a botany faculty member, John Curtis, and a number of his graduate students and colleagues. And so the data that I'm working with are from prairies and savannas and forests. I'm particularly going to talk about the prairie data today, which are in purple. And Laura Ladwig was my postdoc, now at UW-Oshkosh. Amy Alstad was my PhD student. And Dave Rogers is a collaborator from UW-Parkside, and they've all helped with this work. But this is especially Amy Alstad's work. This was her dissertation project. So in tallgrass prairie, we have this really unique system. Just to give some historical context, that habitat was lost following settlement, and so it's in pieces on the landscape and has become even more piecemeal. John Curtis in the 1950s and his colleagues sampled remnants. And then in the 1980s, one PhD student, Mark Leach, resampled a subset of prairies, and we know what the species composition was during that time period. And then Amy went back in around 2012 and did some resampling. So we have the opportunity to look at 3 data points, which is unique, 1950 to 1987, and then 1987 to 2012, to see how these communities have changed. So I'll be showing you these graphs with these 2 time periods, so just so that you have this kind of context. So what this is showing is the annual rate of colonizations and extinctions. The dark bars are the first time period, 1950 to 1987, and the lighter blue bars are the second time period, 1987 to 2012. And there's a couple interesting things about this graph. So this is the rate of colonization per species per year. And the first thing to notice is that colonizations and extinctions are roughly on balance in the first time period. They're both around 40 to 50%. But in the second time period, there's a big gap here between these two bars. So colonization rates are lower than extinction rates. And the second thing to notice is that the rate of change has increased. So the bars are just uniformly taller in the second time period than they are in the first time period. So what this indicates is that there's a lot more change happening in the second time period from the 1980s on than there was from 1950 to 1987. And also where colonization and extinction rates used to be balanced, now extinction rates exceed colonization rates. So we were able to look at a number of potential contributing factors that might influence the rate of colonization or extinction. The first was habitat patch size. So here's a real landscape. It's hard to get at landscape properties in real landscapes, but we were able to measure the area of each of these patches. And we predict that larger habitats would prevent extinctions and promote colonizations. And conversely, in smaller habitats, we'd see an increase in extinction rate and a decrease in colonization rate. So this is again colonization and extinction rates. And on the left is the first time period, and on the right is the second time period. And then on the x-axis, we have patch area. So there was no influence of patch area on colonization or extinction rates in the first time period. But in the second time period, what we saw is that as we got bigger and bigger patch areas, the extinction rate dropped. There was no significant effect of colonizations, even though it looks like there's a trend that direction, it wasn't significant. So the primary thing that we saw was this decrease in extinction rate. So patch area was preventing extinctions the larger the patch got. We also wanted to look at the role of fire. And again, loss of fire is the contributor, or what we predicted would contribute to species loss. So Amy was able to go to all the landowners, and this is where really good records from both private landowners as well as state and federal landowners really has made a difference in the kinds of questions we can ask, because she was able to get these data on the number of burns that had occurred in these different time periods. So in 1950 to 1987, first of all, fire wasn't really happening on the landscape as much as it has more recently, but there was no effect of burn history on colonization or extinction rates. In the second time period, however, the more a site had been burned, the greater the colonization rate and the lower the extinction rate in these sites. So burning really mattered for these, for preventing extinctions and promoting colonizations. So that got us thinking about sort of the interaction maybe between what's happening in the landscape with area and connectivity and the role of fire. So Amy asked this question then about, is there an interaction between connectivity and fire? And so she went to these prairies, and these are data just from 2012. And she asked, what is the total number of species in each of these sites? And then using GIS, she was able to calculate a kind of integrative measure of area and proximity on the landscape of each of these sites to each other. So on the left will be more isolated sites, and on the right, more connected. But just to point out that this is confounding, because this is a real landscape, this is confounding area and any change in patch shape and connection. But it's what we can do at in a real landscape. So just plotting that in general, we saw no relationship between connectivity and species richness, but we predicted an interaction. And so when she split out the sites by whether they had been burned or not, we had these sites that had a maintained fire regime where fire was fairly frequent, and these sites that were not being burned. And you can see there's no role of connectivity when sites were not burned, but there's a really strong impact of connectivity where you get more species when you both have fire and you have this connection. So I think the management implication here is that if you're burning without the connection, you're not going to get as many species. And if you're adding connectivity without the burning, you're not going to have potentially any impact of connectivity on these landscapes, but certainly a less of an impact. Other thing that we were able to look at is community stability. So how much are these communities changing over time? And under climate change, or really any global change, one thing that people are asking is how stable is the community? And so we wanted to ask, does fire increase the stability or the lack of change in a community? So using these same sites, we have unburned in the white bar, low fire, meaning they burn maybe once or twice, and high fire, anything more than that with routine fire. And this on the y-axis is dissimilarity. So it's the amount of change in species composition. So both the presence and composition of these species over time. So here's the first time period, and here's the second time period, and the result is actually the same in both. We see no difference between unburned and low fire, but we see a decrease in the amount of dissimilarity. So this is a bunch of double negatives, but essentially more stability in communities that are burned regularly than in those that don't have fire or have little fire in the landscape. And this is really interesting because it really suggests that fire can be used as a management tool to promote the stability of communities over time. And then I wanted to just also talk a little bit about ways that we can manipulate dispersal besides managing landscapes. And so we have some restoration work, and I think this ties into some of the things you were talking about at the start of this call about local restoration through seed additions and asking how seed additions work across different sites. So coming back to this long-term site, I'm going to only have a few slides from this. I could have a whole talk on it, but I'm not going to today. But I just want to point out that we can manipulate dispersal. So this study was funded by SERDP, the Strategic Environmental Research and Development Program, and done in partnership with Department of Defense, Department of Energy, and the EPA. And we have colleagues at Fort Bragg and Fort Stewart and Savannah River site that have helped with this work. We set up an experiment and we manipulated several factors. We used herbicide as a treatment before we added seeds. We added seeds as a treatment. And we also had these large fences to keep out large herbivores. And it was everything was fully crossed with each other. So we have seed addition with a fence, with herbicide, no seed addition with a fence, with herbicide, all the way to controls that nothing happened. And so we are able to isolate the impacts of each of these factors. What I want to talk about is the role of seed addition. So we collected over 3 million seeds. These were locally sourced from each of the sites. And then we added approximately the same number of viable seeds to each of these plots using identical experimental methods at these 3 different locations. And this is in collaboration with John Orrock and Lars Bredvig. What I'm going to first show you is here's the 3 locations across the top. And the colors will be plots that had seeds added, and in red, and in turquoise, no seeds added. And the first thing I'm going to show is just the mean richness, so the total number of species, and over time. So this is becoming a long-term study. We added the experimental treatments or added the seeds in 2011, and then these are data through 2018. So at all 3 sites, you can see that over time, we actually have generally an increase. There's a little bit of a drop here in 2019, but it's still higher than the initial of seeds, increasing the number of species over time. This is a one-time seed addition. We didn't go back and add seeds every year. So there's a huge impact on species richness just from this one-time addition over the long term. The second thing we asked about was the mean cover of species that were added. So how abundant are they? And in general, we saw an increase in cover even in the last year here. I'm just going to point out that Fort Bragg, relative to Savannah Riverside especially, and even Fort Stewart in large part, this site is relatively intact. So there's a ton of wiregrass in the understory and much less impact of agricultural histories than at Savannah River site. And so my guess is that because it already has a large abundance of these species that we also added, we're not seeing as large of differences. But overall, even the abundances are changing. And this is from a one-time seed addition. The next thing we wanted to ask was just thinking about the key components that we care about in conservation. What's the likelihood of establishment? What's the likelihood of persistence? And then from there, do these species that we add spread out from the plots that we put them in? So here's establishment on the x-axis, and I'm going to plot it against persistence. So persistence meaning that it was there in the last time that we went back to the plots in 2018. And not surprisingly, if the species established, in other words, if that seed germinated and started growing, it was likely to be there in 2018, so many years later. And then we wanted to look at if it established, what's the likelihood of spread? So if it established, and this is spread rates, where we went around each of our plots and walked in meter increments to see how far out they went. And what the important point here is that if they established, they were likely to spread. And any amount of establishment led to high spread rates. So this is the likelihood of spreading. So these are really high spread rates. So what this is showing is that if the species establishes, it will persist over the long term and it will start to spread outward. So in conclusion, just some of the overarching scientific results. From the experiments, we know that corridors do increase species richness through their effect on connectivity per se. And one mechanism that we have evidence for is that corridors do so by promoting dispersal or movement between these patch types. We also know that if you don't have the capability of manipulating landscape level features and you instead decide to manipulate dispersal through seed additions, that you can promote the establishment, persistence, and spread of plants. And then comparing that to long-term observational data from real landscapes, we know that connectivity and fire both increase community diversity together, and they also promote stability. The implications of this for restoration and management is that conservation management that uses area alone, which some tools do that, without considering connectivity, will leave species on the table. So this is not to say that area is not important. Area is the most important thing. But if you only consider area and don't think about the impact of connections between habitats, you will not be able to conserve as many species as if you do. Also, restoring with seed additions indeed promotes establishment, persistence, and spread across a wide variety of different environments. And the long-term observational data suggest that it's really important that these kinds of ecosystems that are maintained in this way with fire, that it's important to consider both the connection at the landscape level and local fire management to prevent species losses. And so, yeah, this is just saying that you need to concurrently plan for that. So I want to acknowledge everyone that has contributed to this and all of the funding that we have received from a wide variety of agencies and programs. Before I finish, I'm going to escape out of this, and I'm just going to put up two things. Just bear with me for 1 second. I'm going to share. I will put the links for this in the chat. Hopefully you can see this. This is a graphic cartoon that Liz Anakozik, who's a really talented artist, made for the results from the paper that we published about the long-term connectivity effects. If you want to share this information and these results with any audience, our hope is that this can help interpret that message. And if I can bring... we also have started a website called conservationcorridor.org. And this is in collaboration with all of the people who are project participants and collaborators on the corridor project, especially Nick Haddad, who is at Michigan State University. But there is a whole host of information here about how corridors work, examples of corridors, and these research digests that take recent research papers and literally digest them to give implications for management and policy. There's different conservation toolboxes and resources. And so if you're interested in landscape level planning, this can be a really good resource. So with that, thank you very much for attending. I really appreciate it. And I'm happy to take any questions. And like I said, I'd also be really interested in hearing what questions you want addressed with research and what you think is important on the ground.
Wow, thank you. That was so fascinating. I was already fascinated by the research you published in 2019, but I really enjoyed seeing the other work that you've done across the nation. And I just think it's just phenomenal. It's really the type of work that we need to do and have people working on. So I appreciate that. We do have a comment in the chat, and also, folks, if you're ready now to go ahead and ask questions, I'll ask you to please raise your hand. I'll read Dana's comment. This is Dana Price from the U.S. Army Corps. She said she wonders if it would be with the same results in an arid ecosystem because they have such difficulty establishing seeded species in restoration projects.
Yeah, well, I totally agree that it's probably ecosystem dependent, but I would also say that, so we put out 3 million seeds and we kept track of how many individuals have arisen. I don't know actually the number that came up, but I do know that it is a very tiny percentage of the seeds that we put out. Interestingly, we have some evidence that, so this is a one-time seed addition. And a few years after we added the species, it happened to be a cool, wet year. And just anecdotally, when we went out to sample, we noticed way more species. So when we were doing our analyses, we decided to ask each year when we had gone back to sample if there was any influence of the climate in that year on the species that emerged, or the abundance and diversity of the seeded species. And sure enough, that comes out as being important. And it's especially important seasonally. So if we get a pulse of rain just before the growing season, followed by arid conditions or more hot conditions, that's bad. That leads to loss, which is obvious, I guess. But if you get more rain in the growing season, then you end up with more germinates. And if the timing is such that you don't have heat after you have this pulse of water and cool conditions going into the growing season, that actually promotes species richness and abundance. So I think thinking through that, and also how climate change is affecting precipitation and temperature patterns relative to germination, is really important. And we're hoping to do some work in that area, pending funding. So I agree with you. And yeah, if there's particular kinds of climatic features in different ecosystems, I think that's really important to consider in comparison to when the species typically germinate.
Yeah, and I think there is a big issue out there too, and she mentions about invasive species as well. So that would certainly be interesting to look at habitats that have been overwhelmed or to various degrees by non-native invasives. I'm also was curious about other disturbance adapted or adaptations, such as things like hurricanes, which you don't necessarily think about as much sort of in the mainland, but it's certainly, you see that happens a lot on islands for sure. And it does happen here, even if we don't think about it. So
right, and the pulse of rain that comes with that, even inland, that's what we're noticing, is that when we have these anomalies and extreme precipitation, it's not so much the wind or like the actual impact of the hurricane, but the precipitation event that happens with that inland is what's seems to be affecting germination. Though this is just from these really kind of coarse data. We'd like to go in and actually put in seeds and look at establishment rates more closely and do some manipulations of precipitation to be able to get at that. And I see that, I think there was a follow-up on that question about, will seeds remain viable long enough to wait? So I guess a couple things. We don't know. And so doing some buried seed bank studies could be good. You can put seeds in window screening and bury them and then pull them out every year to see how long they are viable for. And then also, in a Western system that might be more arid, I am suspecting that there's a lot of annual plants, and it's just a really different kind of system. We're dealing with largely perennials. So when they get established, they're going to hang around. Like, we think that some of these species can live for at least 100 years, herbaceous plants.
Mhm.
So, kind of keeping that life history in mind too, I think is important.
Great. And you see Lori's question there. How many
Yeah.
species were in those 3 million seeds, and how were they decided? And was it
Yeah, I
based
think there
on
were
recommendations?
so there were 21 species, and there's a paper that I can try to find. It's a PLOS One paper with Lars Brodbig as the first author, which was a study that we did before the experiment, where we went and sampled 100 sites at Fort Bragg, Savannah River site, and Fort Stewart to get at the natural range of variation in these sites. So we sampled in burned and unburned on different soil types, in high tree density, low tree density, any variable that we could think of that might be important. We tried to really broadly sample. Then we ran an analysis, a classification regression tree, to just ask what are the groups that come out of this and what are the factors responsible for that. And key was fire regime, tree density, and land use history. So we sampled on sites that had a past land use history of agriculture and sites that did not. And we had sites that had frequent fire using Forest Service or Department of Defense records of fire. We were able to get at that. And so we used that to classify our sites. And then there was one category that was, quote unquote, it's really hard to say, like pristine, best, whatever you want to call it, the ones that were least degraded. And we asked, what are the indicator species using an analysis from those sites? And those are the species then that we tried to target for our seed edition, because those are the ones of most restoration interest for the sites that were the most degraded. We also tried to have species that spanned the full range from Georgia all the way up to North Carolina, or if it wasn't the same species, that there was a closely related congener, because we wanted to locally collect the seed and add it back at those sites and not transfer it, which would also be an interesting question, but not one that we were doing in that grant. And so that's how we decided on the species. And then honestly, what we did was we sent out massive field crews and just asked them to collect as much seed as possible. And we then took it back and we did some initial kind of cleaning and just like seeing if the seeds were whole or not, or if they were empty, and counted, and then used weights. We kind of came up with some rules of thumb there by figuring out how much seed we had. And we went with the lowest common denominator so that the same kind of level of viable seeds was being added to every plot for every species at every location. We also then followed it up with some germination tests for each of the species. So that was included in our modeling.
Okay, fantastic. I see Kimberly has put in a question on whether the pulse rain events are strong enough to actually move the seeds out of the plots.
They could. It's not, there's more vegetation in savanna ecosystems than there would be in like an arid ecosystem. I think you'd have more bare soil in those ecosystems, though that's not my ecosystem, so you can correct me. But so yes, it certainly could. We have buffers around all of our sites too, so hopefully they're not moving between plots at that level. But that's one of the reasons that we also asked about the spatial spread of these species, because it could just be movement out of the plots that way. It also could be reproduction from the plant. So we seeded, plants grew, they seeded, and so it might be kind of like leapfrogging out from there with new plants each generation as well.
Okay, fantastic. Well, we'll take a couple more questions. So Anna asks if the simulated seed germination have you simulated seed germination prior to dispersal? I think you sort of touched on that a little bit, perhaps in the end of this last answer. And have you any observation what portion of the seed was eaten by animals?
Okay, so there we do. So my colleague John Orrock is a plant animal interactions person and specializing in insects and small mammals. So I see both of these questions, that one and the next one about seed predation. His graduate students have done several papers, which I'm not going to be able to recap the high points of those papers. We definitely know that it's out there and it's happening, and we didn't do anything to prevent it for the large scale experimental results, but he had students that put out foraging trays and did some smaller scale work to look at the influence of that, and we know that that's important as well.
Great. And I was going to ask, and I'll let anyone think if there's a last question from the audience. You know, in a way, how much of this is expected given the ecosystems you're working in? And when you hearken back to the old plant ecology type of information about pioneer species and you know, ecosystem climax and things like that, you know, and does it all kind of fit in with what we already kind of know about that?
Right. Well, I guess what I would take there is the corridor project is maybe where you think about succession. So meaning that you disturb and then you watch what comes back. And that project, because of that, it's a reassembly, and we cast it as a restoration, essentially, right? You have a landscape, it's been degraded by past land use and plantation trees, and then management within that context. And so what happens when you cut the trees, you burn, and you kind of start from ground zero? Well, sort of ground zero. First, I'd say that perennial parts, they last in the soil a long time. And if they haven't been tilled in this ecosystem, they're very likely to still be there. So we would, and then I guess get at the effective connectivity relative to succession. If there was no effective connectivity, we just expect species richness overall to do something over time. It might increase over time. It might, you know, have some hump-shaped relationship, but we wouldn't see the difference in the patch types. We'd just see all the patches responding the same. So the interesting piece is when you have different landscape configurations and you're looking at the successional framework, what role does connectivity per se play in that process? I think some of that theory holds up, but some of it might be more, you know, applicable to annual species. And so we quickly got out of that phase. We don't have many annuals in our system anymore. That was really something at the very beginning of our study, and then they kind of dropped out. And so now we have life histories that are mostly perennial, but a lot of herbaceous species. So, and we're not going to go to the climax of, I don't know, some Pacific Northwest forest or something. That's just not the ecosystem type.
Okay, great. Thank you. And let's take a final question from Andrea of, are there any caveats or interesting differences in soil type patch connectivity, such as serpentine and dispersal?
Oh, that's a great question. So I'm just trying to think back to some of the work from the Pacific Northwest. Well, and actually, I'll point you I can send links to this too. I don't know how I haven't had a chance to kind of put these links in the chat yet, but I can when I'm done talking, or I can send them to you. But my student Jesse Miller did some work in the Ozarks that might be the most direct answer to your question, where he was looking at dolomite glades, which is also a special soil, and kind of factors that affected plant community composition there and the role of connectivity. And one thing that he found was that connectivity affects a subset of the population based on when they flower, more so than other species. So there are some nuances there, and it might also be that soils affect the general life history of the species and their dispersal ability because they're able to tolerate soil, and so then therefore they have a whole suite of life history characteristics that are kind of packaged together. And so that's something I'm actually really interested in, is these bundling of life history traits that comes from being on particular soils or in particular burning regimes.
Yeah, I'd be interested in, and I thought some of the work that you did, or it could have just been another researcher, it dealt with bogs specifically. So, you know, where bog can, and they may or may not be connected that we can see, at least with, you know, the watery habitat. And so I think that's always very interesting, because at least 10 years ago, discussions started about rethinking how we frame our refugia, you know, and how we make the boundaries to our reserves and our parks and things, and to take sort of topography and soil types and all that into
Right.
consideration. So I'm
Right.
sure this
And
would
maybe
inform
I
that
think
as
that
well.
more, yeah, the most I haven't done work in bogs specifically, but I think the most general kind of takeaway is, and this is actually where I feel like connections with people who are on the ground. So oftentimes it's manager researcher partnerships that are able to get that kind of really deep knowledge of a system. You have to understand the life history of the species that you're working with to really think about what are the strategies that are going to work. So, for example, if you have a lot of species that are wind dispersed and you have a topographically complex landscape, find out what the predominant wind direction is and think about how topography might be influencing where wind is being routed. But if you don't have a lot of wind dispersed species, then that makes no sense because, I mean, so just, yeah, really thinking about, I mean, in your case, with a bog, you'd be thinking about water dispersal or the movement of water. And so not thinking of connectivity as just like one black box that if you have this structural element in the landscape, it's going to work, but rather how are the organisms using it with their behavior or with the fluid that they're in? How is that moving? And how could you position that to be maximally effective for that group of species?
Yeah, really fantastic. Thank you. Very fascinating. I think we could probably keep talking to you for a while, and I certainly want to keep track of where you're going to be speaking next, because I'm sure every time you'll have some fascinating information to share.
Thank
So I
you.
really thank you for speaking to us
Well,
today.
yeah, thank you for
Yeah.
having me. This has been wonderful. So, and I hope people will reach out. Please email me, or we can set up a time to Zoom. I enjoy making new connections with people and want to collaborate to try to solve these problems.
Great. And if you want to take your time and sort of come up with some of those links, you can send them to Maggie later, and we'll just be sure to include them in the meeting summary
Great.
that gets sent back out. We send it over our normal list, but also to everyone who RSVP, just in case they're not on our regular listservs. So thank you. And I hope you will stay with us for the wrap up and
I
the
will.
next part of our meeting, which I did allow the Q&A to go on quite a bit longer. So we have really just a little bit of time left to morph into the native plants roundtable, which is a time when anybody who's at the meeting should feel free to share information about any events or new publications or updates related to any native plant work that you are involved in. So I'll open up the floor if there are any hands or voices to raise about upcoming meetings or updates on work that you're involved in. Okay, seeing none, I'll have a few. I'll mention that, so my office is the lead in the United States for the Convention on International Trade in Endangered Species of Wild Fauna and Flora, and we just got finished having what seems like a year long, but was actually a month-long meetings, technical committee meetings, to work on specific issues, scientific issues of the Animals and Plants Committee. And it was really productive. We actually discussed a range of issues, including guidance about the term artificially propagated, and a resolution on CITES and forests was discussed. And we also had joint items to discuss with the Animals Committee, such as specimens produced through biotechnology. And for those who aren't aware, CITES, certain species are included in the CITES appendices to ensure that international trade in those species doesn't lead to their extinction. And so that commercial trade isn't driving species to extinction. And so in the US, we do have a lot of CITES listed species that you'd recognize, ranging from Venus flytrap to black coral, the ruby-throated hummingbird, and humpback whale. So I'll share a little more info in our summary notes with that happened with CITES. I want to also mention, I think this is broadly applicable to everyone who's here today, about the America the Beautiful campaign. On May 2nd of this year, the Department of Interior, U.S. Department of Agriculture, Department of Commerce, and the Office of the President issued a joint press release describing a new American the Beautiful initiative with an initial report to outline a vision for a 10-year locally led and voluntary nationwide conservation goal to restore 30% of U.S. lands and waters by 2030. So I just want to put this on your radar screens. There's a lot going on under this initiative. There's a lot that's not figured out yet for this initiative. I know that the U.S. Fish and Wildlife Service is just beginning to build a team that will inform the initiative. And so I'm hoping that we'll have several representatives who from the Plant Conservation Alliance world who can participate in this. Key right now is that they're really trying to engage with private landowners and develop what they're calling an American Conservation and Stewardship Atlas to allow them to self-report on the activities and projects that have contributed to conservation and restoration on their own land. So I think that's a really important component, and I hope that really does engage a lot of the private lands owners and natural resources managers across the U.S. So please just keep your ears to the ground on that. And if you hear that, like I said, this initiative isn't just aimed at federal agencies walking around doing stuff. They're very interested in non-federal partners being involved. So listen up and make sure that our plants, species, and their habitats are represented in that effort. We'll share some links with that. And I don't know if they're shared in the chat now or they might be shared later. But with our meeting notes. I also want to just call out for everyone that there's a global consultation underway on restoration principles. This is to support the implementation of the UN Decade on Ecosystem Restoration, and a task force led by the Food and Agriculture Organization was established to assist in shaping the decade by developing best practices with a best practices task force that partnered with the IUCN and the Society for Ecological Restoration to jointly develop principles that might apply globally to ecosystem restoration. So they're currently in taking public comment and input. And I'm hoping Maggie will be able to post the link to that website into our chat. They ask you to read through, there's 9 principles. I'm not going to read them all. They're too long. You read through that and then you can submit a questionnaire or a survey form. And I hope that you all will do that and just really share your knowledge and understanding of restoration from the plant perspective. There's also a couple of publications I wanted to mention. One is an older one that's actually referenced in, that's been referenced a lot recently, and that is called Estimating the Economic Impacts of Ecosystem Restoration, Methods and Case Studies. It was put together or led by the U.S. Geological Survey and was published in 2016. I do have a link to that as well, and it found that it actually talks about the contributions of restoration to local economies as well. So I think this really gets at the issue that we do need to discuss further on the restoration economy that is part of and that can be built up for ecological restoration. And last but not least, I will mention that for Fish and Wildlife Service, I know that our fiscal year 22 budget has just been put online. It's called the Green Book, and it's part of the Department of Interior budget process. And I encourage folks who are interested in finding out what it is the agencies are talking about doing in the coming years, check out these Green Books, which I think will also be shared in the link, at least for ours. And read about what agencies are doing and learn and consider voicing your opinion on how we get that work done and what we focus on. So with that, can I go ahead, Peggy? I see your little circle blinking. Are you going to speak?
No, I actually was going to ask a question a long time ago about in May, there was the global cactus traffickers are cleaning out the deserts. It was an article in the New York Times. And I was wondering what was going on from the perspective of CITES, because we're having issues on BLM lands with regards to cacti and other,
Succulents.
succulents like Dudleya and all. So
Yeah.
are they gonna do, I mean, because, I mean, we're talking about $1 million worth of cacti on the black market that literally were just whole scale, loss of these plants out of Chile is what the article in the New York Times talked about. So I'm just wondering if CITES is going to do anything on that.
So actually, first of all, if anyone hears about incidents having to do with native species being poached, or, illegally harvested, I'd certainly welcome that information. Not everything's listed under CITES, but cacti, the entire family of cacti and orchids are included in CITES and many other plant taxa. So I've actually been very involved in the Dudleya issue with California. There have been recent poaching events also in Arizona and New Mexico. And so our office is aware of it, and we're actually trying to work with, where we come into play is when those plants go international. And so that's where my office can have an impact. But we're also trying to help coordinate, our local experts, whether there are our Fish and Wildlife Service botanists, our state botanists, with our Fish and Wildlife Service law enforcement and state law enforcement, so that we can try to help coordinate on that. There's a lot of problems in Mexico as well. So we are trying to do things about it. Most of those plants are leaving the country illegally. So that won't have a permit. So, but I do know that it's on the radar screen of our wildlife trafficking program. And so certainly, as I said, if anyone has information, that they, come across, to please do a couple things. You can either email it to me directly, or if it's okay for public consumption, it doesn't have coordinates or something, I encourage you to post it to the PCA listserv and just, help make sure people are aware of these issues. Okay, any other comments or updates or whatnot for the Native Plant Roundtable? I'll mention a couple things. So Society for Ecological Restoration, as I said, we had a pretty strong contingent there from the Plant Conservation Alliance group, federal and non-federal committee. And then I'm sure a number of other folks that I wouldn't necessarily see on this call, but who are definitely connected and doing the good work on the ground. And we'll share information on how that wrapped up in our meeting summary. I also just found out about really late this, another ecological restoration meeting coming up called NCER. I'm looking for it. It is the last week of July. I seem to have lost the link I had up here for it. I don't know if anybody else has heard of that meeting. Let me look here. National Conference on Ecosystem Restoration. It's down in University of Florida. So I heard a few of you talking about Florida today, and it's focused on, I believe, on the US. And so it looks very interesting. I'm going to check it out myself and see if we might not attend. They're still accepting registrations for it. Key among the things that really piqued my interest is that there are funders that we work with that are going to be at the meeting as well. So I think it'll be really useful to check that out and see if you might be interested in participating in that meeting. So I see a note. We're going to just get ready to close up here, but I wanted to. Okay, I see a note from Andrea about the California Native Plant Society has a Dudleya anti-poaching bill making its way through the California legislature. Fantastic. Yeah. And I will put that link in the chat, Lori. Sorry. I'm not good at multi text multimedia. So let us close the meeting by, let me remind you all to please join us at our next meeting, which is September 8th. We'll have Dr. Sam Droege as our presenter, and he's a wildlife biologist with the U.S. Geological Survey, and he'll be talking about replacing lawns with native plant communities, which I think is something we can all wrap our minds around. So thank you all for joining us today. Remember, the easiest way to stay abreast of the work that we're doing is to join our any of our discussion lists. We do focus on different issues in the list, but we also make our PCA announcements and meeting announcements over the list. I posted the link, but it comes up in funky highlight. Thank you, everyone. Thank you, Ellen. Thanks, Anne, for introducing Ellen. We'll welcome you back in your federal hat next time around. And I hope to see you all again. Take care.
Thanks, Patricia.
Bye-bye.